So, welcome back to the immunology lectures, and we will start discussing. So, we started discussing about the complement activation pathways in our last lecture, and we will continue discussing about the complement pathway and try to understand what exactly are the biological consequences. So, what does this complement system actually do, or what are the functions that they perform? So, what we have learned, what we see on the board right now is the complement activation pathway. So, how the whole cascade of complement starts to work, or all the complement proteins, they starts to activate each other and the cascade starts working. So, that was the whole idea. So, what we found in this entire in the complement activation pathways, or what I described in the last class as well, that once there is antigen, so there are three major pathways. There is the classical pathway, the alternative pathway, and the lectin pathway. And the classical pathway starts with antibody antigen binding. Alternative pathway starts only when there is C3b present, only in presence of C3b, the broken product of the C3. And the lectin pathway is primarily, it is also an antigen independent pathway, it is not an antigen dependent pathway. So, the lectin pathway also starts with the interaction of certain lectin molecules, specialized lectins like mannose binding lectin or ficolin, with the carbohydrate moieties on the surface of the pathogen. And remember, all these reactions, most of these reactions in the complement activation pathways, they occur on the surface of the pathogen. It is only on the pathogen surface, and there all of them, if you see, they converge at this point of formation of C3 convertase. So, at least three different sources of C3 convertase we have seen in the last class: the formation of 4B2A both from the classical and the lectin pathways. So, the classical and the lectin pathways are pretty much similar in that way, or their activities are pretty much similar in that way. One is just antibody dependent, another is antibody independent pathway. And then we have the alternative pathway also, where we get C3b-BB. So, breaking down of another factor B and formation of 3b-BB. So, it is 4b2A, 4b2A and 3b-BB. These are the three different C3 convertases, and the main idea is, the central idea is to amplify the signal, and the signal amplification occurs at the level of C3b formation. So, C3b is the main molecule that is required in all the effector paths. So, C3 is broken down by the C3 convertases that are being formed after so much of effort from all these complement proteins. They give so much effort to do one central job, that is to break down the C3. Now, they have broken down the C3 into C3a and C3b. Once C3b is available, this C3b can perform its function in two ways. One is directly, another is again in association with other complement proteins. And what is that? That is, in presence of 4b2a, it can associate with the 4b2a, that is one of the C3 convertase, and produce another convertase which is known as the C4b2a3b, and it is a C5 convertase. So, which means it converts C5 to 5a and 5b. So, these are the two cleavage products of C5. Similarly, this C3b in association with another protein called propyridine can also associate with the C3 convertase derived from the alternative pathway, which is the C3b-BB. and finally, it forms another C5 convertase, which is the C3b, BB and C3b. So, basically this contains 3 different cleavage products, 3 different broken cleavage products, which together they form a bigger complex. And most of these complexes, they are membrane anchored complexes. So, they are anchored to the membrane of the target cell, usually the pathogen, and this is the C5 convertase. So, both of these are C5 convertases, and they can, as the name suggests, they can convert C5 into 5A and 5B. Now comes the main point. So, now we have come to a situation where we have a lot of C3b and we have C5b. We also have C3a and we also have C5a. So, now how all these broken products or the cleavage products of the complement system, they finally mediate the action, or what do they do? So, for that we need to understand the biological consequences or the functions that all these complement proteins or the cleavage products of the complement proteins they do. So, after the activation of the complements, so complement activation, that is what we learnt in our last class, this occurs and this leads to at least 3 different consequences. So, one of the major consequences that we know or we have told or discussed about is formation of the membrane attack complex, or we also call it the MAC. Now, how is this MAC formed and what does it do? We have to understand what this MAC is actually and what it does to the target cell. A second thing is opsonization. And a third thing is enhances inflammation. So, these are the 3 major functions that the complement proteins perform. Among these, this opsonization is an antibody driven process, membrane attack complex. It is primarily driven by other complement proteins. And what are the complement proteins that are involved? The major complement proteins that are involved in this are C5b, C6, C7, C8 and C9. So, now what does this membrane attack complex do and how is this formed? C5, as we discussed in the from the last slide or in the last class, that C3 is broken down to 3A and 3B, and this 3B is the magic molecule. So, this 3B is the central molecule. This 3B can form C5 convertase and it can function independently as well. So, that is, it can have direct action on the cells. So, this is one of the functions by which it has produced the C5b now, and this C5b which is produced on the surface of the cell, it is a cleavage product of the C5, it has formed C5a and C5b. So, once these A are produced, we take this A here. So, C3a and C5a, we put them here because they are the major mediators of inflammation. We will discuss about how they do the inflammation or mediate the inflammation later on, but for the time being let us look into this membrane attack complex. So, what happens is the C5b, when the cleavage product is formed, this 5b is a very labile product, it is not very stable and it cannot survive like that, otherwise it will be degraded. So, it cannot survive like that for more than 2 minutes. So, then it has to be protected or it has to be stabilized, and who does that? So, it is being done by the C6. So, C5 which is broken into C5A and C5B, and this C5B then associates with C6 and forms what we call is C5B6. Now, this is a rather stable complex, because C5 itself gets degraded very fast. So, this cannot survive more than 2 minutes, and now it forms this C5B6 complex. Now, as soon as this 5B6 complex is formed, immediately the C7 comes in and it binds to this C5B6 and forms the C5B6-7 complex. And this C5B6-7 complex is kind of already competent enough to lyse a cell or go into a cell, into the cell membrane. Why? Because as soon as this C5B6 complex is formed and it binds to the C7 unit, then there is a structural transition. So, there is a structural transition, a conformational change, what we call normally in terms of structure. So, there is a quick conformational change, and this conformational change actually exposes the hydrophobic patches or the hydrophobic residues on the C7 on the surface. Now, that leads to imparts more hydrophobicity on the surface and leading to its this assembly of 5B6, 5B6-7 to get inserted into the hydrophobic cell membranes. You know, because the cell membranes are usually, they have a hydrophobic environment because of this lipid bilayers. So, this kind of lipid bilayer is usually present in the membrane, and something that has to go inside this lipid bilayer has to be hydrophobic. Any hydrophilic things cannot go in. They have these lipids, they have a polar head and a non-polar tail, and this non-polar tail or this hydrophobic tail does not accommodate anything or any charged molecules. So, anything that is hydrophobic, that will very nicely accommodate here. So, similarly, this kind of a complex can very easily go inside. Now, sometimes these as I was telling you that most of the time this complement activation process and the assembly of these complement proteins, they take place on the surface of the cell, but sometimes they are also taking place not only on the surfaces, but also in isolated complexes. And if so happens, then sometimes these isolated complexes, which can move around actually, they can go and create the same damage to a cell which is not the target cell, but it is nearby the target cell, and that can happen when this C5B6-7 is already formed. And once this C5B6-7 is already formed, it can go and get inserted into the membrane of a nearby cell which is not the target cell, and this phenomenon is sometimes described as innocent bystander lysis. So, innocent bystander means the one that is standing by, that is standing next to it, it is innocent, but it just gets affected. So, it is just a faulty thing or a mistake of the system that can happen sometime, that it can kill an innocent cell instead of the target cell. This can also happen, and this phenomenon is known as the innocent bystander lysis. This can usually happen starting from this 5B6-7, which is competent enough to go into the cell membrane and can cause the lysis of the cell. So, now the next step is, once this 5b67 is formed, then the next complement protein, the C8, immediately binds. 5b678 complex is formed, and again this complex undergoes a similar conformational change. So, the C8, which is the outermost part. So, they come one after the other and they form a tubular structure like this, and the C8, which is now the outermost one, it can also undergo a structural transition, and then what it happens is it allows binding of C9. Now, the C9 usually binds in huge number, it is not a single C9. So, it can bind like 10 to 15 C9 molecules can bind together and form something what would look like something like this. And a hollow cylindrical structure like this will be formed. Now, this structure can go into the cell membrane, and that would lead to a damage of the cell membrane. So, there will be a hole or a pore formation on the cell membrane, leading to inrushing fluids, leading to disturbance in the tonicity. So, the cells will basically, there will be inrushing fluids and the cells will swell and the cells will finally die. So, they will burst and die. So, what is formed here is C5b678 9, and this is nothing but the membrane attack complex. So, this membrane attack complex, this usually has approximately, the size ranges in the range of 70 to 100 angstrom. The size is approximately in the range of 70 to 100 angstrom, and it looks like a cylindrical structure, and this cylindrical structure gets inserted on the membrane of the cell and forms a channel or a pore and allows the entry of the fluids and leading to bursting of the cell and finally leading to killing of the cells. So, this is one of the way that the cells that the complement system actually can destroy the cells directly. So, after the activation pathway, the final product that is formed is the C5b associates with the C6, and that in turn associates with 7, 8 and 9, and finally it forms the C5b6789 complex, and this 5b6789 complex is known as the membrane attack complex. And this membrane attack complex, as the name suggests, it attacks the membrane, it goes and gets inserted into the membrane and leads to killing of the cell. But usually these events, they do not happen to our normal cells, they usually happen to the target cells. So, the system is very well designed to recognize the target cell only. Sometimes, as I told, it can so happen that these complexes are not formed on the cell or the target cell, and can lead to what is known as the innocent bystander lysis. So, lyse the cell that is standing next to it. So, this can also happen sometime, but usually does not happen, and why it does not happen we will discuss in our next classes. So, let us look into the other process by which complement activation actually shows its effects or its biological consequence, that is by opsonization. So, again C3b, we will come across this C3b molecule several times, because the C3b is the central and the wonder molecule. Now, how does this C3b look like? So, this C3b usually there is an internal thioester linkage by which it can actually have or get an attachment to the cell or the target cell. So, this is how the C3b looks like, and this is the 3b, and this is C3a. So, this portion is the C3a, and this is the soluble part, and this C3a goes here. So, this is the soluble C3a, and this remains bound to the membrane. How? So, consider this as the membrane. So, it reacts with this free hydroxyl or the amine groups, and it can remain bound like this, and this is how the C3b can get bound to the surface of the target cell, if this is the target cell, for example, if this is the surface of the target cell. So, and that is how it can even coat the cell surface. So, for example, if there is a pathogen, if this is the pathogen, for example, a bacteria, it can get coated by the C3b molecules directly by this interaction. And then of course, there are antibodies which are produced. There are antibodies that are produced, and they can also bind to the target cell. Now, this target cell or the target pathogen is then recognized by the phagocytic cells. So, the phagocytic cells or the phagocytes, so let us call this the phagocyte. These phagocytic cells, they can recognize this by specialized receptors like the complement receptors, the CR1 for example. They can recognize by these complement receptors, as well as there are these Fc receptors which can bind to the Fc region of the antibodies. So, when there is these two reactions occurring together, so when there is an antibody binding to the surface of the target cell, as well as C3b molecule, which is also known as an opsonin, because of its opsonization function, it is also known as an opsonin. So, it is primarily known as the opsonin. So, this kind of C3b molecules, they coat the surface of the pathogen, they coat the surface of this pathogen, and as well as the antibodies which are formed, these antibodies, they also coat the surface of the pathogen. So, when these two interactions are complete, that leads to internalization or phagocytosis. So, this makes phagocytosis complete. So, that would lead to finally internalization and phagocytosis. So, that is how the C3b, actually the C3b molecule mediates opsonization. So, the C3b molecule, usually the C3, the entire C3, it can remain bound to the surface of the cell, and when there is a cleavage of this C3 by C3 convertase for example, that would lead to C3a and C3b. The C3a is usually the soluble part, and the C3b is the insoluble part, because it remains bound to the membrane. So, now this C3b can other than leading to formation of C5b and membrane attack complex and all these things, the C3b can directly coat a pathogen surface. It can go and bind to the surface of the pathogen by this kind of a linkage shown here. So, it can go and bind to the pathogen surface, and as well as the pathogen or the bacterium, it can get coated by the antibodies, and together they can bind to the receptors on the surface of the phagocytes. So, the CR1, the complement receptor, or the FC receptors, and these FC receptors can bind to the FC region of the antibodies. So, when these two types of bindings are complete, then this phagocytosis can occur, and this cell is then internalized and killed. A third process that the complement proteins initiates, or the cleavage product of these complement proteins initiates, is enhancing inflammation. So, they can bind to receptors that are expressed by this type of cells, specialized cells which contains these granules or the granulocytes, and binding of these receptors, binding of these complement cleavage products can lead to degranulation. We will study more about degranulation in our next classes. So, that leads to degranulation and releasing primarily histamine and other mediators of inflammation, like for example prostaglandins. So, this process is known as the degranulation, and this is primarily mediated by the soluble cleavage products or the smaller cleavage products like the C3a and C5a. It can also lead to, if you remember one of our very initial classes where we studied about the inflammation or the inflammatory responses, and we have described about the extravasation of the neutrophils, for example. So, the neutrophils, the extravasation of neutrophils, that also, or migration of the neutrophils, that can also be mediated by the cleavage products of the complement cleavage products or the soluble cleavage products like C3a and 5a. So, these can also act as chemoattractants, and this can also enhance inflammation. So, they can bring more and more of these neutrophils, more and more of these phagocytes, the monocytes, they can come to the site of action. So, that leads to enhanced inflammation. So, they can either lead to degranulation and release of the histamine and other mediators of inflammation like prostaglandins, or they can also assist in migration of the leukocytes, migration of primarily the neutrophils to the site of action, and by that they can lead to enhancement of inflammation. So, what we learned in this lecture today is that the complement system mediates its action. What we learned in our last lecture is how the complement system gets activated, what are the major activation pathways, how they cleave each other and gets activated. And what we learned today in this lecture is that how these activated complement proteins, or the after activation, how cleavage of these proteins can lead to finally what are the biological consequences of this activation of the complement pathway, and they can kill or destroy the target cells. So, one of the ways it does is by formation of the membrane attack complex, the MAC. A second way it does is by opsonization along with the antibodies. It helps the antibodies as well. So, along with the antibodies, it helps in engulfment of the pathogen or phagocytosis of the pathogen. And the third is it can enhance inflammation. So, these are the three major ways that the complement proteins actually performs their function. Now, the question is, so if these complement proteins can do so many things, so many harmful things, why don't they do it to our self cells, to our own cells? It targets the pathogen. It only creates membrane attack complex on the pathogen. It only opsonizes the pathogen or coats the pathogen surface. So, why or how this, when there is no infection in the body, the complement proteins are still there, the complement proteins are still moving around. So, why does these proteins, even though they are moving around in the body, they do not really attack the cells of our body, or they do not really do anything, any inflammation or anything. So, why this is happening? Why the complement proteins do not perform these functions normally or do not get activated normally? So, of course, there is a reason, and the reason is that there is a very tight regulation. There is a very tight regulation, and there is another class of proteins which are known as the regulators of complement activation, the RCA. So, this class of protein, the regulators of complement activation, this class of protein, they do not allow the complements to, even if there is a cleavage, even if there is a breakage and formation of C3b, availability of C4b, even then there are regulators which will not allow the complement to perform all these functions, or the complement system to perform all these functions, and they are known as the RCAs, or the regulators of complement activation. We will discuss about the RCAs and the complement complex regulatory processes in our next lecture. So, that is all for this lecture, and thank you very much.